Mitosis. During interphase, the nuclear envelope, represented by the orange Sour Patch Kids, encompasses the nucleus, represented by the green Sour Patch Kids. The nucleus has one or more nucleoli, represented by the mini Reese's cup. The yellow Sour Patch Kids are the centrosomes with the centriole pairs.
In prophase, the chromatin coils and condenses into chromosomes, represented by the 3 different pairs of colored gummy worms in the nuclear envelope. The duplicated chromosomes have 2 sister chromatins that are fused by a centromere and connected by cohesins. The mitotic spindle begins to form, which is represented by the red Twizzlers. The mitotic spindle is made up of microtubules and centrosomes. As the microtubules lengthen, the centrosomes move away from each other.
In prometaphase, the nuclear envelope breaks and the microtubules extend from the centromeres and enter the nuclear envelope. At this point, the chromosomes are tightly compressed to each other. A kinetochore develops on each of the 2 chromatids of each chromosome. A kinetochore is a specialized protein located at the centromere. Some of the microtubules attach to the kinetochores. The attached microtubules jostle the kinetochores from side to side.
Metaphase is the longest phase in mitosis. The centrosomes are now located at completely opposite ends of the cell. A metaphase plate is an imaginary plane that runs through the middle of the cell, dividing the spindles equidistantly. The centromeres of the chromosomes are positioned on the metaphase plate.
During anaphase, the kinetochores of sister chromatids attach to kinetochore microtubules for each chromosome coming from the centrosomes.
In telophase, 2 daughter nuclei are formed in the cell. A nuclear envelope is developed from the fragments of the parent cell's nuclear envelope and other particles of the endomembrane system. Nucleoli begin to appear, the chromosomes become less compressed, and mitosis is completed. Cytokinesis, the ending stage of mitosis, is the division of the cytoplasm. Cytokinesis begins to occur during telophase and is completed shortly after. The result is 2 separate daughter cells. The cutouts of the plate represent the beginning stages of cytokinesis. The cleavage furrow, which develops in animal cells, can be seen by the black line drawn across the plate. It pinches the cell, helping to divide it in 2.
Meiosis is a 2-part process consisting of meiosis I and meiosis II. Meiosis I, the separation of homologous chromosomes. In prophase I, the chromosomes begin to compress, homologs begin to align by genes, and crossing over begins. Synapsis ends by mid-prophase, and each pair of chromosomes moves slightly apart. Each homologous pair has 1 or more chiasmata, and the homologs are still associated due to cohesion between sister chromatids. The nuclear envelope disperses, spindles form, and centrosome movement occurs. In the later stages of prophase, the microtubules from each pole of the cell attach to the 2 kinetochores, and the homologous pairs move towards the metaphase plate. The cell has 2 sets of genetic information, making it a diploid cell. Diploid cells are represented by the number 2n. In this cell, 2n equals 6.
In metaphase I, the homologous chromosomes line up on the metaphase plate. The chromatids of one homolog attach to the kinetochore microtubules of one pole, and the chromatids of the other homolog attach to the microtubules of the opposite pole.
In anaphase I, cohesion separates homologs along chromatid arms, and the spindle apparatus moves the homologs towards opposite poles.
Telophase I and cytokinesis occur simultaneously and form 2 haploid daughter cells. In the beginning of telophase, each half of the cell has a complete haploid set of replicated chromosomes. In animal cells, a cleavage furrow is formed, but in plant cells, a cell plate is formed. Replication does not occur between meiosis I and meiosis II.
Meiosis II, the separation of sister chromatids. In prophase II, the spindle apparatus forms, and the chromosomes move towards the metaphase II plate.
In metaphase II, the chromosomes position on the metaphase plate and the 2 sister chromatids of each chromosome are not genetically identical because of the crossing over that took place in meiosis I. The microtubules extending from opposite poles attach to the kinetochores of sister chromatids.
In anaphase II, the proteins that hold the sister chromatids together at the centromere break down, allowing the chromatids to separate. The chromatids then move to the opposite ends of the cell as individual chromosomes.
In telophase II and cytokinesis, nuclei form, the chromosomes begin to decompress, and cytokinesis occurs. 4 daughter cells are produced from the meiotic division of one parent cell. Each daughter cell has a haploid set of unreplicated chromosomes. Each of the 4 daughter cells is genetically distinct from the other daughter cells and from the parent cell.